Alright, so DNA. Basically, it's the genetic material that makes up who we are individually. Everyone has different genes, which means they have different DNA to each other. So, I'd have different DNA to you, basically. And DNA is found in the nucleus of a cell. So, I'll draw it for you. There's the cell, and it's got like the little ribosomes around it, and the nucleus is in there, which basically inside that has the DNA or the chromosomes that have DNA in it, which basically are X's, and inside that they have the little DNA strands, which obviously they look like a double helix, which is also described as a ladder shape. So basically DNA looks like a ladder when you unwind it. It's basically, usually it looks like that, but we display it as that for basic things, obviously. DNA is made up of many little molecules, and that makes it a polymer. So basically, DNA on its own is a polymer, and the little things on each strand of DNA makes it up each monomers. So basically, what DNA is made up of is a phosphate group, a deoxyribose sugar, and a nitrogen base, which they can either be adenine, thymine, cytosine, or guanine, which basically adenine pairs with thymine, cytosine pairs with guanine for reasons. Basically, each strand of DNA is made up of little monomers, which is basically the sugar, phosphate group, and the nitrogen base, and they end up forming a ladder kind of shape. So basically, basically they form a ladder shape, which kind of looks like this, just not so good here because I'm trying to rush. And yeah, so what connects the little DNA molecules is bonding. There's two types of bonding. There's covalent bonding and hydrogen bonding. Basically, covalent bonding is on the single strands. It connects each little nucleotide together to make the whole strand, and that bond is very strong as it is sharing of atoms in both molecules. And then hydrogen bonding is between the actual base pairs, which is where on each of the bases, one of them has to have a nitrogen or oxygen, and the other one has to have an hydrogen. So basically, the hydrogen either connects to a nitrogen or oxygen, and then that forms the hydrogen bond. But that bonding is very weak, so it's not so good. But it is good for when it's strong enough to keep it together, but it's good when you want to do DNA replication, which basically you just use the enzymes to break it up, add the nucleotides to it, and then you can form a new DNA strand or new DNA piece from your original piece. So basically, yeah, the base pairs actually have a specific shape. There's two different types of shapes. Two different types of base pairs. There's purines and pyrimidines. Purines are cytosine and guanine. Oh no, guanine and adenine actually. and then pyrimidines are thymine and cytosine, which they are different. They don't actually pair together. They're the non-pairs. So what I'm saying is adenine is not paired with guanine. Guanine pairs with cytosine, obviously, and AT. Yeah, and then they have different connections, different numbers of bonds or hydrogen bonds in them because of their actual shaping and where their molecules are on the base pairs. So basically, hydrogen bonding in cytosine and guanine, there's three hydrogen bondings. So basically, that means there's on one of the sides there's either two nitrogens or one nitrogen, one oxygen, and one hydrogen, and on the other side there's two hydrogens and one either nitrogen or oxygen, and then basically that pairs that, and then with adenine and thymine there's only two bondings, which basically similar kind of problem. And yeah, so basically the shaping of the pyrimidines, they've got two rings, and one is basically shaped like the sugar and one's a octagon. So basically pyrimidine kinda looks like that, where a purine just looks like sugar basically. So basically it just looks like that one. So that's pyrimidine, that's purine. And yeah. So also DNA, as I said before, DNA can be replicated. So basically that's just splitting the two strands or unwinding them with an enzyme, which is pretty sure DNA helicase, I think it is, which splits it up, and then one strand goes normally, like in DNA there's primes, and they connect 5 to 3, so the 5 prime connects to the 3 prime, so I'll show you. Basically the phosphate is the 5 prime, and then just the connecting bit is the 3 prime. So DNA strand goes down this way on that side, and then on the opposite side the other DNA strand goes the other way as it is upside down, and that's the way it connects. And yeah, but yeah, it splits. DNA splits by DNA helicase. Then it's either a group of DNA fragments or base pair fragments or connect, or nucleotides connect, or single nucleotides connect, and they're usually connected by ligase, I think it is. And then the other strands, there's something uncommon with that, or unfamiliar, I mean. It doesn't connect properly because of the backwards, or the wrong direction, the two-direction DNA kind of thing. So basically, it's a lagging strand, so it doesn't go one way. It doesn't go like that, and it basically you need to use Okazaki fragments which help connect it properly, but sometimes there's unusual things that don't work out with that. So yeah, and then you can also split up your DNA into little pieces, and you can use it to for DNA profiling, which you can use gel electrophoresis. So basically, you cut up your DNA pieces, or with what's called with um, with you split it up with stuff, obviously. And then you place it into these wells. So basically, it's a square or a rectangle. Actually, it's a rectangle. There's a positive end to it and a negative, and it's actually upside down because the phosphate group in DNA is actually negatively charged. So yeah, I'll explain that after. But yeah, there's wells here, so you have like 3 or 4 wells, and you match it up to whatever. Say it's a crime, you have the fingerprints or the DNA from the fingerprints here. It'd be like that, and then you have like 3 suspects, and then like you see their DNA splitting. So one there, one there, one there, and whichever one matches it is the right criminal actually. So the way the DNA moves down the wells is because of the phosphate group in the nucleotide. It actually it's negatively charged, so it moves towards the positive. And the larger the pieces, the smaller the pieces actually, the further they go. So the smaller the pieces, the smallest piece will be down the bottom, where the largest piece will stay at the top or very close to the well. So yeah, hopefully that's alright and I get past with that, hopefully. And sorry about the mishap last week. Couldn't really help it, but yeah. Cheers. Tim.